Okay, hi everyone. Um, just to confirm, can you hear me? Like thumbs up, is everything fine?
Yes,
Okay.
we can.
Okay, cool. Then yeah, let's start with that presentation. I'd like a deep human proteome sequencing for global detection of mutations and alternative splicing. This was a collaborative project between Joshua Kuhn here in Madison and Jürgen Cox in Max Planck Institute in Munich. And I was a PhD student doing this work actually while being in Max Planck Institute and already as a postdoc in medicine. And this was a very fruitful project, and I hope I kind of encourage you to read this paper and think slightly different about proteomics after that. But first of all, I would like to apologize that I didn't come to Boston in person. And I have to say, I have so many friends in Boston and so many cool activities in life science happening in Boston that I really wish to come, but not that time, unfortunately. And the reason is actually the reason is that. So I'm starting from 17th of June 2023. I become a father, and so this is I call this call of duties, called for duties, fatherhood. And it's a lot of fun, but unfortunately we have a few dicey weeks right now, and I just feel it's very inappropriate to leave my wife to deal with this diciness. So yeah, but thanks for opportunity actually to present this via Zoom, and this is actually a very excellent opportunity. Okay, cool. The thing is that my talk gonna be focusing on different degrees of freedom, which we are usually not capturing with proteomics. So let's say from one end, from genome annotation, we know we have 20,000 genes. And as we obviously know, almost every gene results in a few different protein variants, different, let's people call proteoforms, or let's call protein variants. And this is a huge, actually, space of different proteoforms. And this space is constructed thanks to the many different degrees of freedom, which makes actually 10,000 genes into the many, many different proteoforms. And these degrees of freedom, such as alternative splicing, such as non-coding mutations, and as well as all kind of post-translational modifications. And as we know, we can study with top-down proteomics this variety which result like variety of different variants of proteins. However, not in the large scale, not in the scale of all, let's say, 50,000 proteins or even more. So, but as we know, one of the way of studying actually this complexity, this is a shotgun proteomics, when we actually taking the protein, digesting on a peptide, and trying to kind of investigate what's going on with all this variability. But however, as you also know, shotgun proteomics results in just a few peptides per protein. And in order to actually detect all these variations, which are very sequence specific, we need almost 100% sequence coverage. And that's what's the focus of our work. But let's go slightly slower for that. So let's just take the typical experiment, protease digestion with a trypsin. We're taking some random protein, and trypsin generates many different peptides. And as you see, miscleavage, all these peptides have miscleavage 0. However, you have to also take into account that sometimes it can be miscleavage 1 and 2. However, out of all this space of potential peptides, mass spectrometry, LC-MS system can detect just a subsequence of that. And there's usually like just a few peptides which are suitable for sequencing. And this is due to the different properties of the peptides, right? Like length of peptides, charge of the peptides, hydrophobicity of peptides, etc., which makes more or less suitable for actual MS analysis and detection. But okay, now we see that using the trypsin, we can cover approximately 20% of all amino acids of that specific protein. But how can we increase this coverage? Like, let's say, amino acid coverage. And one way to do so, we can just take another protease, right? If you take, let's say, glu-C protease, it will generate slightly different set of peptides, and the subset of these peptides will be detected with LC-MS system. And we hope that this peptide is going to be slightly different compared to the peptides which we detect in trypsin. And if we combine all these peptides together, we just boost the coverage, amino acid coverage of this protein. And with hope that that's going to be useful for detecting some small sequence variants, which we're going to discuss further. Yeah, cool. And in order to do so, we decided like to boost everything in terms of numbers. Instead of taking one cell line, we took like 6 cell lines. And I just like to note that this is exactly the same cell lines which have been used for ENCODE project. Next, each cell line from each of the pilots of the from each of the lysate of the cell lines, the proteome have been extracted, and all these proteome mix have been digested with different proteases, actually 6 different proteases, and it was digested separately for each protease. Now we have 6 multiplied by 6, and each of these protease digest have been fractionated, extensively fractionated up to the 30 fractions. And each of the fraction have been measured with like tribal system, and thanks to the tribal system, we can actually use different fragmentation methods, HCD, ATD, and CAD. And all this data set have been analyzed with MaxQuant, but I would like to just emphasize how huge this data set is. So if we multiply all these combinatorics together, 6 cell lines multiplied by 6 proteases multiplied by, in average, 25 fractions, multiplied by 3 different fragmentation methods, it's going to be approximately 2,500 files, which is 5 terabytes of data, and which is just half of the year measuring time on the instrument. It's a obviously huge data set, and it's analyzing this data set itself was actually a huge development effort of the MaxQuant in the first place. But we made it, and actually through the review process, one of the reviewers said this paper describes a technical tour de force that generates a useful resource. Indeed, it is, and this is a DDA data, and it generates like millions of different annotated spectra, and what's actually cool about this data set, it's actually amino acid coverage. That's what's the purpose of this generating such experiment. And let's take a look at the one example, hemoglobin subunit gamma 1. Obviously, this protein is highly expressed. Obviously, it's going to be biased in terms of detection rate, let's say. But if you look at all peptides which we detect from this protein and map back on the sequence and count how many peptides covering each position, each amino acid in this protein, we'll find out that there's some positions which have more than 9 unique sequences, unique peptides. But in average, it's true, in average, it's around 5 peptides. This is awesome, right? So now we are talking about proteomics data like we're talking about transcriptomics data. We do have 100% coverage for some proteins, obviously not for all, but and that's kind of opens opportunities for many questions which have not been answered before. But let's go through the data a bit deeply. Like let's discuss it like through different angles. Like first angle is let's take a look from actually standard definition of protein depth. How many proteins out of 100% we're detecting? And as you see, like we're taking like different cell lines or cell lines all combined, and we are kind of distinguishing between how many proteins we detect with such protease and such protease, or all proteases combined, and which is a gray circle. And we do see that trypsin outperforms all the rest of proteases, and that's true. Trypsin generates really nice peptides with arginine lysin at the end, which already generates like positive charge by default, and the length is more or less suitable for the mass spectrometry. But the truth is that usage of the rest of proteases brings a lot of additional information, and that's kind of what the quintessence of this. It's actually amino acid coverage. How many amino acids out of 100% of amino acids in a human proteome we're covering with tryptic peptides, or with the rest of all the rest of the protease peptides. And we see that trypsin generates approximately 1 million unique peptides for the trypsin, unique amino acids, covering 1 million of unique amino acids. However, if you take all the rest of proteases combined, that's going to be approximately 2 millions. And overall, if you take all proteases together, we are covering approximately 8 millions of amino acids. And this is quite significant, which I believe never been reached before, even like in the data sets where people combine many different data sets. from community. The truth is that also, if you take trypsin, and for each gene, we calculate sequence coverage, which is on y-axis, and rank sort all these proteins based on the sequence coverage from the lowest to the highest, we find out that this distribution looks like that, which is no surprise, let's say, more or less normal distribution across sequence coverage. But however, if you take all the rest of proteases on top of the trypsin, and which is represented by gray dots, we see that every gene, every protein, like the sequence coverage of this protein in terms of unique peptides, amount of unique peptides, amount of covered amino acids, boosted up to 20%, plus 20%. And that is great. And but from one point of view, you can say, yeah, like using just trypsin, it's actually simple. And you're suggesting right now using 6 different proteases, which is just increased complexity of sample preparation, etc. And that is true. And if, let's say, if you would like to go for some subset of proteases, which increase the coverage up to a certain level, you can look at this actually figure. So first of all, there's 2 bars for each of the combination of proteases. The first dark gray is actual observed coverage distribution, and the light gray, it's theoretical. If all tryptic peptides have been detected, then we will detect 92% in average, in median, coverage for all proteome. And obviously, like there's a theory and there's a practice which is far away from the theory. However, we see that trypsin alone outperforms all the rest of proteases. But if you suggest to use just 2 proteases, then we kind of see the coverage more or less the same, 62%. But all these proteases include trypsin. And just addition, but you see, just addition of any protease except trypsin will, like addition of any protease on top of the trypsin will increase the coverage from 54% to the 62%, so by 8%. And of course, like if you take all possible combinations of 3 proteases, it's going to increase again, and etc., etc., etc. And the truth is that the median coverage of that dataset, which we measured, is approximately 80%. So over all proteins which have been expressed in these cell lines, we detect that median coverage over all these proteins is 80%. This is enormous. Cool. But there's also problems, which I feel like everybody have to be aware to be very kind of scientifically rigid to discuss how useful this data set. So one of the problems which I would like to highlight, which is actually well known in the field, it's transmembrane proteins. The transmembrane proteins are notoriously difficult target for shotgun mass spectrometry, and we kind of also observe in data set the same. We also can make the same conclusion from our data set. So if we plot distribution of the coverage for proteins without transmembrane domain or with transmembrane domain, I will highlight quickly. If we plot distribution for proteins without transmembrane domain or with transmembrane domain for different proteases, we see that the proteins with transmembrane domain have, in average, have lower and actually statistically significant lower coverage, and for all proteases separately or for all proteases combined. And yeah, from that moment you can say, but there's no surprise in that, because transmembrane proteins usually are low expressed proteins, right? And that's why we observe lower coverage, because lower expression generates less amount of peptides in the case of DDA experiment or any experiment actually. Yeah, but the truth is that actually this observation is not about transmembrane proteins as a whole proteins. It's actually about transmembrane domains. And the thing is that we see that this drop of the coverage, it's a relative coverage normalized by -15 position from amino acids N-terminus or C-terminus. We see that this coverage dropping down immediately when we come to the transmembrane domain, or this coverage increased when we are leaving transmembrane domain. So that just suggests it's not just about transmembrane proteins because we have a low expression, but it's actually specifically about transmembrane domains which are very difficult to sequence or cover. And there's no surprise in that, right? Because these transmembrane domains are extremely hydrophobic, and as a result, we usually don't have charged amino acids, and it explains trypsin, but it also explains the behavior of chymotrypsin. We see that chymotrypsin relatively performed the best out of all different proteases, which makes sense because chymotrypsin targeting usually hydrophobic amino acid. Cool. And I feel like this very important to highlight this. And it's like maybe in future we find out a better way to deal with transmembrane proteins. But for now, this is like Achilles knee, like in terms of coverage. Okay, cool. But with this all huge data set, which is 80% median coverage, what can we do? The first thing which kind of come up in our mind that we can actually detect single amino acid polymorphisms. It's basically nucleotides change, let's say from ATG to AAG, which involves just changing in one nucleotide, which result changing the whole amino acid. And the question is, can we detect such amino acid exchange on a whole global scale? And will it be coverage of 80%? Will we detect actually 80% of all single amino acid polymorphisms which we detect in transcriptomics level? And the reality, yes. We extracted polymorphism not from genomics data, but actually from transcriptomics data. As a result, we are sure that these mutations are expressed. And in average, for almost every cell line, we're detecting approximately 80% of all mutations on the proteomics level. And that is great. That means that and there's no surprise that it's 80%, right? Because we saw that median coverage 80%, then if these mutations distributed randomly across whole proteome, then we also should detect them with 80% coverage. And but at that point, we were kind of honestly slightly been shocked because no single study was showing 80% detection rate of all single amino acid polymorphisms which we detected on transcriptomics level. And that level, we were kind of slightly skeptical, is it true or not? And actually, to convince ourselves, we come up with different quality control metrics. And one of the metrics was actually Pearson correlation coefficients between actual predicted spectra from deep mass and actual observed spectra, but observed from reference peptides without mutations, and peptides with mutations. So basically, let's say we're taking peptides with without mutations, we're predicting spectra, intensities of different fragments, and we're correlating these two things. And we're doing this for many actual reference peptides, not for all, but for many. And next, we are taking the mutated peptides and generating spectra for them, and also correlating with observed spectra. And the thing is that we're not actually interested in actual value. Is it high or low? But what we're actually interested in, how similar this Pearson correlation coefficients between these two types subset of peptides with mutation or without mutations. And as we can observe, this value is actually very similar. And after that moment, we actually were way more convinced that these peptides are correctly identified, correctly annotated. But another thing which we kind of have been curious about, but is there any difference in terms of gene sets for genes where we're detecting mutations and genes where we don't detect mutations? Is there any Go enrichment in another term? And as a result, we again observed exactly the same outcome as before, that undetected mutations or genes with undetected mutations are heavily enriched with membrane parts. So all these dark blue, navy color dots, actually, this is Go terms with a membrane in it. And we see that this is probably the most significant enrichment factors between genes with genes where we detect mutations on transcriptomics level, but we don't detect on proteomics level, compared to the genes where we detect on transcriptomics level and on proteomics level. Which no surprise again, right? Because we remember that membrane parts of the proteins actually notoriously difficult to sequence. Yeah, but thanks to actually reviewers who forced us to look at that, we found out we constructed a small hypothesis which actually we're very much in favor to check very, very soon. So, and this hypothesis was such that imagine that why we don't detect certain mutations, even though we see some mutations on transcriptomics level, why we don't detect them on proteomics level. And one of the many, let's say, one of the many potential reasons why we don't detect such mutations can be that mutations actually killing protein. It's making protein unstable. As a result, this protein destroyed. As a result, expression of this protein either reduced to zero, and that's the reason why we don't detect this mutation. And in order to prove that, or let's say to strengthen our hypothesis, let's put it like very smoothly, we choose two softwares like SIFT and Polyphen. It's a software which taking mutation information and predicting and scoring each mutation, how badly it is for overall for physiology of the cell. It's rated mutations from deleterious to tolerated in one case, or another case is probably damaging and benign. And this is, I have to highlight this, softwares have been used extensively for exome sequencing and for prioritizing different mutations for being damaging or not. And we decided to make this prediction for a subset of mutations which we do detect, and subset of mutations which we observe on transcriptomics level, but we don't detect them on the proteomics level. And what we found out that the subset of deleterious or probably damaging mutations actually increased in a subset of undetected mutations, which actually indirectly and very kind of weakly, let's say, supporting our hypothesis that these mutations, which we don't detect, actually very dangerous mutations, where increasing instability of protein, we're killing protein, and as a result, we are unable to detect these mutations. And that is actually very interesting. But again, this is very much hypothesis, and we need to make additional work to prove that, which actually we are working on right now. And the second part, which I believe one of the most exciting part, this is detection of alternative splicing. But believe me or not, this actually was a huge question. Is there any alternative splicing products on a proteomics level? And you may know that people from transcriptomics world will tell you for sure there's a huge amount of alternative splicing. Take a look at the different mega project, ENCODE, TCGA. Everywhere we do detect many different alternative splicing events. However, the question is this splicing events, splicing variability translates to the proteomics level. It was really open question. And I feel like this is actually really ridiculous from my point of view, but that was how it was. Imagine that, let's just introduce what's alternative splicing, right? It's like there's a one exon which ends up with codon, which translates in amino acid A, and there's a in between exon, which translates in two amino acids, and there's a last exon, which translates in amino acid S. And exclusion of this exon results in AS, or inclusion will result in ASMS. Yeah, the slide from ASMS. But what I would like to highlight today is actually, was already shown before, but we kind of rediscovered this, that trypsin is not the best protease suited for study of alternative splicing. And this was slightly shocking, but this is due to the actually very intrinsic properties of splicing and actually trypsin itself. So, but let's come up with this conclusion like smoothly. So if one will come up with idea of measuring distance in terms of base pairs, in terms of nucleotides, I'm sorry, from 5' of exon to the N terminus of peptide, you'll find out that most of these distances actually are equal 0 or 1 or 3. It's also true for, if you look at the distance between C terminus of the peptide to the 3' of the exon. And usually it's either 0, either 3. It's actually very specific pick for lysine here, but probably be discussed later. In conclusion, we observed that the peptides which we detect, especially for trypsin and lysi, and actually indirectly for the lysine, where frequently either start at the beginning of the exon or ends at the end of the exon. this is actually a very painful observation, because these peptides are non-informative to distinguish one alternative splicing from another alternative splicing event. And that's why trypsin is not the best choice for the study of splicing, splicing in general, or alternative splicing. But in the same time, right, you can ask, but why do we observe why do we have such a strange observation, right? Why do we see this peptide starts exactly at the same time as a 5' of exon, or ends exactly the same place as 3' exon? So the distance between peptide and end of this exon is usually 0. But the truth is that if you know how splicing works, there's a kind of certain recognition patterns, certain sequences which splicing machinery recognize and splicing in. But the truth is that some of these recognition patterns actually expand on the coding sequence. And believe me or not, this after removing the exon, intron from the premature mRNA, the sequence which kind of frequently loves to be there, it's a codon of lysine amino acid, lysine or arginine, even like that. So it means that lysine or arginine really loves to be in between exons, in between exon junctions. And that's why trypsin generates many peptides which end or stops synchronously with beginning and end of exon. Quite annoying, right? But that's how it is. But from another point of view, that's the power of this method, right? The power of this study, where we used many different proteases, except including like trypsin. Okay, but next I would like to introduce you like definition. First of all, I would like to show you the example of alternative splicing on actual protein, and next introduce a few definitions, which we're gonna use later. So imagine, it's actually true, there's amyloid precursor protein, and inside of this amyloid precursor protein, there's exon 7, 8, and 9 on genome level. This gene can be translated on, can be transcribed, and the premature RNA will appear with the same sequence of the exons. But next, thanks to the alternative splicing, exon 8 can be excluded, and then it's going to be called exon skipping, or exon 8 can be included, and then it's going to be called no skipping. And this is kind of called alternative splicing exon skipping events. And we believe, like naively first, that this variation on transcriptomics level, we will translate it on proteomics level. That's in the case of proteins, it's going to be exon skipping again from 7 to 9, or no skipping, then it's going to be 7, 8, and 9 exon. Cool. But then we're plugging in our data. We're plugging our results from our project. And what we do detect, we detect that we detect many peptides, let's say 3 peptides here, which span between 7 and 9. And as you see, there's a many different amino acids on which lies on the exon 7, and many different amino acids which lies on exon 9, which just support that we are correctly assigned this peptide to this splicing event, splicing alternative. Or we also see many peptides which supports either 7 to 8 exon jump, exon junction, or 8 to 9. And obviously, all these peptides supports no skipping event. And sometimes, obviously, both these alternatives can coexist in the cell line, and that's actually what we observe frequently. But as you can imagine, like each of these peptides have been detected, have been fragmented, and we have usually very nice spectra for each of this peptide, and we are very much convinced that this peptide, the spectra is not fake. It's actually went through the very robust statistical analysis. Cool, but you see, one thing which we were kind of very curious about, that is this alternative splicing prefer one alternative, either just exon skipping or no exon skipping, or both together. So when we are saying that we're fully detecting alternative splicing event, it means that we're detecting some peptides which supports exon skipping or supports no skipping, both simultaneously. Or when we are saying partially detected alternative splicing event, it means that we either detected some peptides which supports exon skipping, either no skipping, but not both together. And that's kind of what we are showing here. So, okay, first of all, I show you example of skipping exon, right? But as we know, there's a way more different type of alternative splicing. And this is alternative splicing, which is skipped exon, retained introns, alternative donor, alternative acceptor, or mutually exclusive exons. This is a subset of 5 alternative splicing events, which do have names. I have to kind of like to highlight that there's many alternative splicing events which are way more complicated in terms of graph structure, but we don't have names. And actually, this is I think one of the gems which I discovered through doing this project. But anyway, so first of all, we took the annotation which is available on Ensemble website, and this annotation tells us that exon skipping is the most popular way of doing alternative splicing. Next is alternative acceptor, next alternative donor. But out of all these 12,000 events, we are partially detecting, at least partially detecting 9,000 on transcriptomics level, and 4,000 where we detect both alternatives being present in the cell lines, which is 4,000. And in the protein level, of course, numbers are way less compared to the transcriptomic level, but it can be different biology or detection limit. But we do detect, partially detecting 3,000 exon skipping events and 700 where we detect both alternatives. And that's actually very excites us because we kind of now with all these like numbers, we actually can ask some very interesting questions. So and make some interesting discoveries. And one of the unexpected discoveries which we made, this is related to the out-of-frame alternative splicing. And we can declare that out-of-frame are not present on a protein level. But what is actually out-of-frame alternative splicing? Let's look back on our example, like the simple exon skipping event in frame. In case of in-frame, as I said, we have exon 1, we have exon 2, and exon 3. And this exon 2 is actually variable. It can be excluded or included. But it's actually important that exon 2, the length of this exon is either 3, either 6, either 9, either 12. You got the point. It should be divisible by 3. So as a result, inclusion of this exon will not introduce frame shift for the ribosome, which is going to work further. But in another case, imagine opposite out of frame case, where this exon, variable exon, the length of this exon is not divisible by 3. Let's say in that example, the length of this exon is actually 5, but this can be also 4, this can be also 7 or 8, but not 6 and 9, right? So as a result, inclusion of this exon will introduce a different frame of translation for the third exon. And usually the situation result in the case when there's an early stop codon which kills one of the alternative, which kills one of the splicing alternative. And that's actually what we observe in our dataset. If we highlight the alternative splicing events which we have fully identified or partially identified, or in frame or out of frame, we see that the alternative splicing events which are fully identified almost exclusively kind of in frame alternative splicing events. And we almost don't detect anything in the case of out of frame in terms of fully identified events, which just actually means that that's one of the alternative out of 2, which kind of result of this alternative splicing is actually destroyed. It's actually non-functional, or it's maybe even unstable. And that's why we never ever, almost never observe peptides which cover both alternatives in the case of out of frame events. Cool. Another thing, like as you see, right, we discussed one factor which impacting our capability to detect or not detect certain alternative splicing event. And one of the factors you see that's this exon which is excluded or included is a in frame or out of frame. In other words, is this exon, length of this exon is divisible by 3 or not divisible by 3, right? But at certain moment we realize that there's actually many factors which are more or less expected or non-expected, which impacting this probability of being detected or not detected on proteomics level, even though all these alternatives we detect on transcriptomic level. And what we decided to do, we decided just to take all these factors, one by all these factors together, and ask machine learning to actually binary classify alternative splicing events which we detect with proteomics and which we don't detect with proteomics. And the thing is that we don't care so much about this usage, like application of this binary classifier. What we are actually more curious about, we're curious which factors machine learning model highlights as most important. And that's what we did. We used XGBoost model binary classifier to distinguish between alternative splicing events which we detect on proteomics level and which we don't detect on proteomics level, fully detect or not fully, or not detect at all. And we found out that if you plot the rho curve, this is approximately a UC of 83% thanks to the cross-validation of sevenfold. But again, what was more important for us is actually to find out what's importance of different factors which impacting this detection. And we found out that most important three factors are transcript abundance, percent spliced in, and frame status. The frame status we discussed with you. That's exactly like being out of frame or in frame, being divisible by 3 or not divisible by 3. But transcript abundance and percent spliced in, some two new parameters which we were not aware, which we are kind of hypothesized before, but actually it's very cool that machine learning highlights these two factors. But I will prove you that actual transcript abundance and percent spliced in is just two different sides of the same coin. First of all, let's discuss transcript abundance or overall gene expression. If we, again, bin this transcript abundance by 1 ppm, so we find out that overall, the rate of full detection of alternative splicing events is increasing if you go to the range of the high expressed genes, high expressed in terms of transcription in that case. And if we take the highest abundant proteins or genes, which have transcription abundance 7+ RPKM, which is actually logarithmic RPKM, which is really enormous, but we do detect them. So we do detect 50% of alternative splicing events on proteomics level, where we detect both alternatives. But yeah, again, you see there's a very clear pattern: higher expression, higher detection, higher chance to detect alternative splicing. And it's no surprise, right? We know in any omics this bias. There's an omics in transcriptomics, in single cell transcriptomics, in proteomics. There's an obvious bias towards highly abundant genes. We do have a coverage for the highly abundant genes higher compared to the low abundant genes. It's just a matter of the detection rate. But another thing, it's actual percent spliced in bins. The factor of percent spliced in, or PSI, in the term which is actually very much famous in the transcriptomics world. But what does it mean? Let's say percent spliced in. So percent spliced in is the ratio of included exons, like out of 100% of gene expression, how many, what's the percent of the alternative splicing results in inclusion of the exon out of 100%. And that's what's actually called percent spliced in. And let's say example of 90%, right? It means that 90% of alternative splicing events results in inclusion of the exon and 10% in exclusion of exon. And the truth is that if you think about this, like we already saw the problem with gene expression, right? Like the high abundant genes tends to be more well covered compared to the low abundant genes. That's no surprise. But now, if you take such genes which have PSI value of 0.9 or 0.1, you're basically reducing initial gene expression in 10 times for one of the alternative. So basically 90% of the gene expression results in one alternative and 10% in another alternative, but we actually would like to detect both. And that's actually frequently a huge factor which defines, because this is basically diluting initial gene into the low expression. And as you see, that's actually just another side of the same coin, which called expression of genes impacting probability of being detected, impacting probability to detect alternative splicing. Cool. But at the end of this talk, I would like to highlight one important message. And you know, it was a huge discussion during the writing of the manuscript, and in general during the revision. Like reviewers ask us, can you please prioritize which factors out of proteases, out of fragmentation, out of fractionation, which of these three factors impacting most of the on the amino acids coverage. Like if I would have very limited amount of time, I don't have the time to vary all these factors. What would be the best factor to actually to start with? And we at that moment we can obviously say this is a usage of multi-protease. It's the fact. It's the one of the most influential factor which impacting the amino acid coverage. And that's true, like not even for alternative splicing. It's true for any splicing sites, which obviously happens frequently. That if we again plot percent of identified splice sites based on transcriptomics coverage, we see that all proteases, all proteases combined, will increase at certain moment, but at certain moment we're gonna go on plateau. And but the truth is that all these proteases have more or less similar, let's say 20% plateau. But one of the way to increase this plateau is actually usage of many different proteases all combined. And this is, we believe, can even further improved. We believe that this 40% ceiling can be even like increased higher, but that's what we are working on right now. But I believe at certain moment we'll be able to reach like unbelievable 80-90%, and then we will be freely speaking about alternative splicing events, and that would be awesome world of 100% coverage or near 100% coverage of proteomics data. Cool. But let's say if you have some specific gene of interest and you would like to know which non-sequence mutations we detect there, or alternative splicing which we detect there, you just can go for the deepsequencing.app application, the website, and spike in your gene name and find out what we actually detected from this data set. This basically doesn't require to load the whole data set, which is huge. It's like a fast screening for gene of interest. The whole story have been published recently, and actually very much proud of this paper because it was a huge collaborative effort of many different groups all around the globe, which was really hard to organize in terms of Zoom meetings. But this was actually very exciting. And I have to say, I learned really a lot about alternative splicing while doing this project. And frequently it was unexpected results. And I feel like it's very cool. But the rest of my time, honestly, I would like to just to talk briefly what's next. How this project going to evolve further. So in order to start talking about that, I would like to highlight that obviously this is very this is very difficult experiment to do because this is there's so many combinatorial factors such as many different cell lines, many different proteases, huge amount of fractionation. It's like altogether it creates a huge amount of samples and huge amount of runtime. And obviously what we are and I also would like to highlight this project was mostly focusing on identification rather than on quantification. And this is due to many things, but I feel like next step, which we would like to go further, it's actually pushing direction of quantification. There's many ways doing so. First of all, we can reduce combinatorial explosion, right? To use many, not use like 25 fractions, but use, let's say, 4 fractions, use some subset of proteases maybe, but we kind of think about this right now, and actually we are in process of doing so. But I hope soon we're going to come up with some exciting model where we can apply quantitative approach. Will it be DTA or DIA? It's actually an open question yet. So we will try out and find out the best solution for that. But that's going to happen soon. And another thing is mutations. And I would like to very much to continue this effort of finding out that certain mutations are killing proteins or not killing proteins, but that's probably will require some targeted approach. We will find out soon. Yeah, and I would like to thanks to Joshua Kuhn, who hosted me like in this in medicine today as a postdoctoral researcher. It is an awesome group, very much like focusing on hardware development and software development and sample preparation. And obviously, I would like to thank Jürgen Cox, who was my PhD supervisor and like supervisor from the Big S. And I really thank for inviting you for Max Mann Summer School, and I'm ready for questions.
Can you talk about how you did the searching? I mean, you couldn't use just a single UniProt database. How did you end up doing the searching to find these places and know, like especially when the sequence exons are missing in the middle?
Yeah, that's a great thing. So we use not just okay. If you use UniProt file, right, you have to use both. You have to use UniProt with standard Swiss prot, like standard gene to protein one ratio file, but also you have to use additional file, which contains all type of isoforms. But still, if you have this UniProt 2, even 2 UniProt files, you will slightly struggle with some cases, struggle to translate these proteins into the genomic sequences, unfortunately. But as a result, we decided to use a UniProt file plus additional and standard UniProt file, plus we use ensemble faster files, which kind of translated open reading frames for from a UniProt annotation. So the thing is that you may say that this will explode amount of different proteins, explode amount of different peptides, but it's actually not true. Max won't take care of that. He collapsing proteins which have very much similar peptide sets into the protein groups, and we actually resolve this issue well. So yes, you have to use many different FASTA files. At least you have to use both UniProt files, standard and plus additional, and yeah, that's how we dealt with that.
So I was curious if you've considered doing like a co-digestion with single cells, or if that would be too complicated, the mass spec results of that. Like, you know, digesting a single cell with multiple sort of digests, right? And seeing what trying to analyze the spectrum from that, or if it's just been like on bulk cells, like in this case.
Okay, so I feel like there's 2 questions here, right? First of all, single cell, or it was combined, right, in complicated way, but single cell, or, and the second question about multiprotease digestion, like doing adding different proteases in the same sample. So first of all, like single cell, I don't believe we're actually ready for that yet. So the thing is that even fractionation step will require a lot of material to start with. Probably soon it's going to be miniaturized, that we'll be able to do all these things together, but not now, I would say. I would be very curious, but cautious to say this is possible. But second thing is combining different proteases or combining different digests, right? So combining different proteases is probably over difficult because this will generate, you know, combining different proteases. Yeah, you can combine lysine and trypsin. That's fine. That's actually standard approach. But combining, like, let's say, trypsin and chymotrypsin in the same tube, that's probably generate huge amount of peptides and probably very small peptides. I would not recommend to do so. But what you can do, you can actually combine different digests. Let's say you made trypsin digest, you made a chymotrypsin digest, and in order to save time, you just can combine different digests in tube, right? And the thing is that the like first author of this paper, Alicia Richards, she actually published a really nice paper on that topic. She, in order to reduce amount of effort, reduce amount of measuring time, she combined different protease digests and measured this complicated proteins peptide set. And there's advantages, disadvantages as usual, right? But I would just to highlight there's one huge disadvantage, that the search space will be like increased or doubled, right? And there's some probability of misinterpreting different digests. So it can save time, but it can increase headache. So, and that's what actually we're going to do right now. We're going to try to explore what would be the best combination of proteases, what would be the best maybe combination of different proteases or digests, sorry, or it can be even different like acquisition methods, right? DDA or DIA. That's what we are working right now. Thank you for the question.
Thank you for the very nice talk. So you have 5 or 6 cancer cell lines, but the reference data set that you are using are from the healthy samples, UniProt or Swiss prot with alternative splicing. In the previous Max Quant summer schools that I watched online, there was a discussion about the dependent peptides. Is that an option that you can use?
Okay, I would like to first of all clarify one thing, that we used mutations from transcriptomic data, right? And we enriched our database with alternative splicing, may think, from annotation, which doesn't cover the case when some mutation happen on translational level, right? And it doesn't cover the cases when some completely new alternative splicing can happen, or some crazy modifications which we are not including in database can happen. That's true, and that's what like when we are talking about that, that's a dependent search. It's this data available. Everybody can do that. We haven't done that, and the reason is because we were kind of trying to narrow down the story to the publishable state, because everybody needs to have deadlines, and that's what we did. But I feel like it's possible, and I feel like if you're curious enough, I would, in your case, try that. And I feel like it's actually the whole story by itself, and I encourage you to do that actually. But we haven't done that.